guys, my name is Michelle Fu. I'm a WPTO Marine Energy Summer Intern on the Seedling Project, and I will present the goals and preliminary results of the project. So Seedling projects are part of a program with DOE Water Power Technology Office, which supports new research in the field of marine energies. And as an intern working on the Seedling project, I already presented a version of today's talk during the WPTO Seedling Water Power Showcase last Wednesday. Okay, so this seedling project led by Lene Henry in the Coastal Sciences Division aims to assess the use of environmental DNA or eDNA to monitor changes in marine habitats caused by offshore energy developments. So monitoring surveys can be expensive in high energy environments targeted for wave, tidal, and offshore wind energy. And these environments have especially strong tidal currents or high waves. And the question we wanted to address was, could we use water samples only to survey these habitats and detect species of interest using eDNA methods? And with this project, we are testing the possibility to use eDNA to survey fish and invertebrates at a tidal site. And to do so, we are collecting water samples and comparing the results to underwater photos and assessing the costs and methods of collecting and processing eDNA as compared to surveys by beach sign nets or scuba divers. Okay, so what is eDNA? We like to think of eDNA as like a marine version of CSI, where in the show, the characters use DNA and other evidence to piece together a crime. Here, eDNA uses DNA in the form of discarded detritus, such as fish scale, skin, feces, and blood in order to piece together the identity of either a single organism or even like whole ecosystem-wide assemblages. And so outlined here are the general steps for performing an eDNA survey. So first, eDNA is sampled, and eDNA can be sampled from marine environments such as deep sea environments and coral reefs, freshwater environments such as lakes and rivers, and even from soil and benthic environments. And in our case, we are interested in investigating eDNA in marine environments. So next, the water samples are filtered in order to concentrate the genetic material. And lastly, a chemical process, usually extraction kits, is used to extract the DNA from the filters. So why should we use eDNA? And there are several advantages to using eDNA surveying when compared to conventional survey methods. So unlike visual surveys, which often need like specialists to identify species correctly, eDNA surveying does not require specialists to perform surveys. And eDNA surveying is not as invasive as other catch-based methods like trawling, which often destroy benthic environments. And lastly, eDNA can detect more species at a higher resolution. And this is really because the genetic material stays in the environment for some time after shedding. And so even if the organism is not present anymore, it can still be detected. So like when compared to limited time conventional surveys, eDNA provides a more complete picture of the species found in that area. And furthermore, in the lens of surveying in high energy marine environments, in order to quantify the effect of marine energy technologies on the surrounding biodiversity, conventional surveying methods have limitations. So in our case, for CNET surveys, which involve setting up nets to catch and identify fish, rough waves or a strong current can make it impossible to sample. And so sampling opportunities have to be limited to when waves are calm. And in addition, the handling of organisms may cause stress and ultimately increase mortality rates, while some quicker and smaller species may also escape the nets. For visual surveys by scuba divers, rough water conditions may also make sampling unsafe as well as decrease visibility. And scuba surveys may also be prone to detection bias and error. And eDNA can circumvent the problems faced by CNET surveying and scuba surveys. So advantages of using eDNA can be shown in the field data. Lene collected water samples in March, May, and August in the tidal channel of Squim Bay, and she filtered the water samples on site at PNNL and sent the filters to a subcontractor for sequencing 3 genetic markers targeting fish, invertebrates, and macroalgae. And here I present you the preliminary results regarding the fish species identified in the May samples, and the red dots show the sequences that were also found in the March samples. Buffalo sculpin was the only fish species observed by the scuba divers during sample collection, but the eDNA results showed that many more benthic species like gunnels and flounders and pelagic species like herring and salmon use the habitat in the channel. And from these results, eDNA methods seem to provide a good representation of the biodiversity in the tidal channel. And like reported in the literature, the methods allowed the identification of more species than observed by scuba divers. Okay, so for my portion of the seedling project, I conducted a literature review to find the average cost of conducting an eDNA survey, which then can be compared to the cost of CNET surveys and scuba surveys. And I did this through keyword searches in online databases, and I reviewed over 200 peer-reviewed articles. And from the gathered articles, I extracted data such as brand and type of container used for sampling, filter type, and whether a pump was used for filtering, DNA extraction kit used, etc. And shown here is an example of the extracted data as well as the type of paper we looked at. So after extracting the information from the articles, I would search for price estimates for each item. And when possible, we use several websites to construct an average price for each piece of equipment. So for example, shown here is part of the spreadsheet I used to organize the cost. So when a paper specified that they used, let's say here it says 2-liter Nalgene bottle for sampling, I would look up 2-liter Nalgene bottle and write down the name of the product, the group it belongs to, whether it is part of sampling, filtering, or extraction. And next, the cost per each was found. For example, on a pack of filters, we would write down the cost per filter. And lastly, we would note the link. And pieces of equipment that were no longer available to purchase were omitted from cost analysis. Okay, so here are some preliminary results. For sampling, we looked at deployment methods as well as type of collection method. Some studies use boats and others sampled from the shore. So noting whether a boat was used for sampling is important, as there are various costs associated with operating a boat, such as fuel, labor hours. And for our results shown here in the pie chart in the top left, we found that sampling events were performed with and without a boat in equal measure. And we noted NA if the paper was unclear about whether a boat was used. And we also looked at the type of collection method used as shown in the pie chart in the top right. And the most common type of collection method was with a bottle of unspecified brand, which was followed by a container of an unspecified brand. And the next most popular collection method was Nalgene brand bottles. Because using simply bottle or container for cost analysis is not that helpful, I opted to use Nalgene brand bottles for cost analysis. And here I didn't include a cost estimate for each type of collection method because there is such a large range of costs associated with each type of collection method, depending on the size of the instrument. So as such, for our final cost analysis, I will use a range of costs, for example, helping construct a lower cost and a higher cost for collection methods. So in terms of the filtering step, I found that the most common pore size was 0.45 nanometers. I also looked at whether a pump was used, and a peristaltic pump helps pump the water through the filter, usually in the context of filtering on site or in the field. And while a majority of studies did not utilize peristaltic pumps, about a fourth did, and thus a cost with and without the pump will be computed. And lastly, I looked at the average cost of each type of filter, and the upper and lower bounds of each bar in the bar graph shown in the bottom right represents the most and least expensive filters in each category. And so here we can see that the cost of the 0.45 nanometer filter is highly variable, and this is due to the large number of brands of filters for this particular size, and in particular filter capsules, which were on average more expensive. They were only using the 0.45 nanometer pore size. So without the inclusion of filter capsules, the average cost of filtering for the 0.45 nanometer pore size would be $2.34 per filter, which aligns with the cost of other pore sizes. And so lastly, I looked at the most common DNA extraction kit used, and the most common extraction kit used is the DNeasy blood and tissue kit, which the average cost for each preparation or sample is $3.54. Okay, so not only did the results regarding sampling, filtering, extraction reveal the most common practices and the equipment used in each category, but my literature review revealed the diversity found in the eDNA procedure. So in total, I found 3 types of deployment methods and 56 unique methods of collection for sampling, and 17 different pore sizes for filtering, and 19 extraction kits or methods of extraction. And beyond the metrics that I quantified, other variables that we did not explicitly bin also varied for each step. For example, variances between studies in terms of filtering and extraction included the volume of water filtered, as well as the preparation steps of the filters for extraction. As, for example, in some papers, the filters would be shredded prior to extraction, others did not. And this diversity in practices and materials really reflects the fact that eDNA sampling is a relatively new field in which the best practices are still being explored and consistency in protocols remains to be achieved. Okay, so as my internship was extended for 2 more months, the results above are preliminary and the next steps are to finalize the overall cost analysis and to compare to eDNA costs and eDNA costs. So I will accomplish this first by setting up case studies with predetermined parameters such as time and space to compare the 3 methodologies. And this is because several factors such as labor costs and the cost of operating a boat, for example, they are dependent on the time and location associated with sampling. And once the parameters are determined, I can then compare the costs. And here below is a list of materials and considerations for each type of sampling. These associated materials and costs for each conventional surveying method were obtained from discussions with PNNL researchers who are subject matter experts. Okay, so I would like to thank Lene on guiding me on this project thus far and helping me structure my project, as well as Nicole Sather for also guiding me, especially on the topic of CNET surveying. And thank you for listening to my presentation, and I'll open it up to questions.
Thank you, Michelle. So we have a question from Claire. What was the most difficult part of performing this cost analysis?
Yeah, definitely. Like I said before, there is such a great diversity in the methods associated with eDNA. So I guess a big a difficult part of performing this cost analysis was really finding papers that would check the a good amount of the bins that we constructed. So for example, we would have papers that would tell us absolutely nothing about what filter pore size they used or how they even sampled, and that wouldn't really be helpful to our analysis. So we would have to maybe not use that paper. So we would go through and try to find papers that did check off all the things we needed. Yeah, great question.
Yeah, great question, Claire. All right, Michelle, thank you. Way to recover. And next up, we have Liahi Josens. You can go ahead and share your screen and go ahead with your presentation.
Great. Okay, hello everybody. Thank you for coming to my talk today. My name is Liahi Josens, and I'm a SULI intern working under the mentorship of Dr. Allison Myers-Pigg within the Earth and Biological Sciences Directorate at PNNL.
Liahi, you're sharing your notes screen.
Oh, shoot. I'm going to redo this.
Display settings. I think it's just.
Oh, here we go.
Yeah.
Now, better?
There we go.
Okay, so try this again. Hello, everybody. Thanks for coming to my talk today. My name, or actually, I already did that, so let's just go into the normal part. So today, I will be presenting my research on an investigation of soil water soluble organic carbon in relation to depth in salinity. So as many of you may be aware, as our climate changes, we expect to see an increase in the magnitude and frequency of storm surges, as well as rising sea levels, both of which will lead to an increase in salinity exposure of soils in coastal ecosystems. My summer SULI project was a much smaller subset of a larger project, which will be occurring later this month. It is called Tempest, and it's a large-scale flooding experiment that will occur in situ on the soils of an upland coastal forest. So basically, large volumes of both freshwater and seawater will be dumped onto plots of land in order to see how things such as vegetation and carbon dynamics change and respond to the floodings. So my SULI project gives a small glimpse into what we might expect to see in the larger scale flooding experiments. Albeit my experiments were done in a laboratory setting rather than in situ and solely focused on water-soluble organic carbon. So first off, what is water-soluble organic carbon? A very general definition of it is that it's a carbon stock, a mobile carbon stock residing in soils that can move from terrestrial to aquatic ecosystems through mechanisms such as flooding. Using this diagram in the upper right, we can see that carbon contained in soils can start off as water-insoluble organic carbon, and then this can then be transformed into via processes such as degradation, desorption, and leaching into water-soluble organic carbon, all of which can occur in aquatic environments. So down below here, you can see a picture of the soil solutions from which we extracted the water-soluble organic carbon for my SULI project. This carbon is of interest to researchers because of its participation in the global cycling of carbon, as well as its connection to both chemical and biological processes occurring in soils. So for my project, what we did is that we were interested in seeing how soils taken from the same coastal ecosystem as the tempest plots would respond to freshwater versus seawater exposure. In particular, what my project did is that we exposed soils from 7 different depths, which you can see here on the right, to both freshwater and seawater treatments in order to test our 2 hypotheses pictured here. So we hypothesized firstly that seawater would extract less water-soluble organic carbon than freshwater due to the effects of polyvalent cations such as calcium and magnesium. And then secondly, we hypothesized that saltier water would extract less aromatic organic matter due to higher ionic strengths. The parameters that we looked at were bulk concentrations of magnesium and calcium, as well as concentrations of DOC. And then finally, we looked at aromaticity of the DOC via specific UV absorbance at 254 nanometers, which can be used as a proxy for viewing organic matter aromaticity. So as you can see in this graph here down below, aromaticity is positively correlated with higher SUVA 254 values. Aromaticity is of interest to myself and other researchers because of its connection to biodegradability and bioavailability of soil organic matter. In general, aromatic organic matter is generally less or more resistant to biodegradation by soil microorganisms. So our experimental procedure was as follows. We took soil samples from each of our 7 depths and then exposed separate sets of samples to freshwater in triplicate and then seawater in triplicate. After exposure to each treatment, the samples were filtered to remove the particulate soil matter contained in the solutions, and then the remaining solutions were analyzed for each of the parameters of interest, which you can see here on the right. Please note that we use dissolved organic carbon as our representative for water-soluble organic carbon. So going straight into our results, what we found is that bulk concentrations of both calcium and magnesium were near 0 for calcium and then 0 for magnesium in freshwater treated soils, which are in red, and that you can see towards the bottom of those lines towards the bottoms of the graphs. So we found that magnesium and calcium were significantly higher in the seawater treated soils, with highest concentrations occurring in the 10 to 20 centimeters range. So the variability in bulk ion concentrations for both calcium and magnesium are attributed to each ion being dominantly present in the seawater rather than in the soil itself, seeing that the freshwater treatments had such relatively low or near 0 amounts of calcium and magnesium detected in the soil solutions, in the freshwater soil solutions. So please note that we purposely did not blank correct our sample so that we could see the ions contained in the seawater. Next, looking at water extracted DOC concentrations, we found that between the two treatments, there was no significant difference found for concentrations of DOC in the surface soils of 10 to 20 centimeters, but that in general, fresh water extracted significantly more water-soluble organic carbon than soils that were treated with seawater. Note that all these samples were blank corrected to account for background carbon signatures contained in the seawater. As you can see, there is a really large difference between the concentrations of DOC detected at the surface relative to deeper soils. This is to be expected, although it would be interesting to get a more comprehensive analysis of the first 20 to 50 centimeters, just to see to what degree concentrations of DOC are changing in that first half meter of soil. So relating these results back to our hypotheses, we did see a correlation between the amount of polyvalent cations present in bulk soil solutions and the amount of dissolved organic carbon extracted from soils. Put simply, the freshwater solutions, which did not contain significant amounts of each cation, extracted more DOC than the seawater solutions, which did contain high concentrations of each cation. Other researchers have attributed this correlation between the parameters to the coagulation that can occur between cations and soil organic matter. Basically, in the presence of certain ions, organic matter will coagulate, making it less soluble, and therefore leading to lower concentrations of water-soluble organic carbon that can be extracted from soils. Our final parameter of interest was to look at aromaticity of the carbon contained in the samples from each treatment. So our second hypothesis was basically that freshwater would extract more aromatic water-soluble organic carbon than seawater across all depths. We did not observe this trend. Instead, we saw that freshwater had lower levels of aromaticity above 120 centimeters, and then higher levels of aromaticity between 120 to 500 centimeters. We had originally thought that we'd find correlations between ion concentrations and the aromaticity of soil organic matter, but overall, our results are pretty variable and leave a lot of room for further experimentation. So for future directions, there's room for further analyses on the remaining ranges of depths that we did not get to. That would be like the depth on the far right, the soil depths pictured in that kind of light tan color. This would give us a more comprehensive study of the carbon dynamics of the soil profile. Additionally, I began running some biodegradability experiments towards the end of my SULI term, the results of which did not make it into the final product, but it would be interesting to see how biotic factors such as microbial populations contained in seawater may affect and contribute to soil carbon dynamics, specifically in regards to bioavailability and biodegradability. Finally, it will be interesting to see how the results from my SULI project compare with the in situ results collected at the Tempest sites when they are flooded towards the end of this month. I'd like to acknowledge that this work was supported by the DOE and its subsets. And I'd like to give a huge acknowledgement to my mentor, Dr. Allison Myers-Pigg, who's been an incredible teacher and source of help both inside and outside of the lab. I'd also like to thank my colleagues at Mackerel. Thank you for sharing the lab space with me, answering all of my questions, and just being an overall fun group to work with. Finally, I'd like to thank Nicole, Juan, and Brandy, and the entire Office of STEM Education for all their help in making the SULI program a success. So with that, are there any questions? Or thank you, and are there any questions?
Thank you. Yes, we have a question in the chat box. I'll go ahead and read it to you. It's from Eve. What differences might we expect to see in this project between lab and in situ settings?
So I think that's a great question. I think in the in situ settings, like results and also parameters looked at will be more robust. I think in the like in the in situ setting, they're going to be looking at vegetation dynamics as well. So I think they'll just have a lot more data and also more analyses. And so I think that it'll just have more to go off of when they finish all of their sampling and all of that.
Good, great question, Eve. All right, since we're running a little bit behind, if there are additional questions, please put them in the chat box. And so we'll continue with our next presenter. Ariana Moore, please go ahead and unmute yourself and share your screen. And you've got the floor. There you go. Can you hear me? Okay.
Hello, my name is Ariana Moore. I'm a PhD intern here at PNNL. I'm part of the GEM program. I matriculate into my program at Georgia Tech next week, and I'm part of the environmental subsurfaces group here. My project is database for efficient cataloging of cementitious waste form properties to enable development of a machine learning tool. So right now there are around 56 million gallons of nuclear waste stored at the Hanford site. I have a picture of the interior of a waste tank on the bottom left. The Hanford Waste Treatment and Immobilization Plant is intended to treat this waste via vitrification. Vitrification means to make into glass, and the picture at the upper right is an example of such waste glass. The glass provides a long-term stable form before final disposal. However, during the vitrification process, secondary wastes are produced, which also need to be immobilized before they can be disposed of. As a potential solution to this, PNNL and Washington River Protection Solutions, as well as other laboratories across the nation, are working on a grout development program, which is intended to produce cement-based waste forms for waste immobilization. At the Hanford site specifically, we'd be using these waste forms for immobilization of those secondary wastes from the vitrification process. But there's also a potential for cement waste forms to be used as a supplement for low activity waste immobilization. And the reason for this is because 90% of the waste at the Hanford site is low activity waste. So it would just help with the large volume of low activity waste. On the right are images of the various cement waste forms. While these on the outside look like typical concrete cylinders, they're actually engineered with different properties, and those properties change depending on what's put into the cement waste form. So using grout formulations or using cement formulations for waste immobilization isn't new. There's actually decades of existing literature. However, right now there is no central location for cement waste form data, whereas there are several databases for vitrification. Right now, the assumption is that researchers would have to individually read through each paper to extract whatever the necessary information that they need. And that's really an inefficient process, especially if you're thinking about this as a potential large-scale waste management option. So that's where my project comes in, which is to create a database to serve as that central location for grout formulation data. The overall goal is to utilize a database to capture data and allow for the development of an artificial intelligence or machine learning tool, which would help predict what the cement waste forms design and performance would be kind of on the site as operations are going based on the continually changing chemistry of the waste. But that's a very large project, so I've kind of split it up into a overall picture of how we're basically funneling all this data in to compress it all down to this usable AI program. My elements of the funnel I've put are in the asterisk. So I'm the part who's actually creating the database structure and doing a lot of the database testing and feedback. What was really important is that the database needed to be user focused. It needed to have detailed quantitative data so that you wouldn't need to go read through the papers. It needed to maximize data quality, and most importantly, it needed to have a flexible data structure because the needs of the research group would inevitably change over time, and they wanted a database that could accommodate that. So there are a lot of elements to the database, but I essentially separated them into different functions based on their worksheet tabs. So first, there are two types of data entry. Then the actual data is split into two sheets. So one for grout formulation and performance testing data, and the other one is for liquid waste data. Then the export and visualization sheet gives users the ability to pull out data based on keywords or data values. Lastly, for a lot of the features across the database, Excel actually needs a place in the background to store and save certain settings. And so I've essentially put all the background operations in a background sheet, which only admins would be able to see and have access to. So again, because the database is very large, I figured an easier way to talk about it is to walk you through what type of data we store. So as already mentioned, there are two ways to enter data. The first is a sample by sample data entry form. And this requires users to enter these 5 types of data, starting with the report information. Then we have the sample information, which includes assigning sample IDs, saying what type of waste the sample is containing, and what the makeup of the waste form is. So the sample IDs are numeric, which is a level of standardization for this database, because individual papers use all sorts of methods for how they name and number their samples. And then there's a sample ID note section to make it easier for researchers to connect the database samples to the samples in the individual papers. Next is the physical performance data. These are characteristics of the waste form itself. It includes flowability, or how easily the cement mixes and pours into the waste molds, how long the cement takes to set and harden before it's ready for disposal, and how strong the cement is when it's under pressure. Next is the immobilization performance data, which is the most important part for a lot of researchers, as it helps them determine how effective the cement waste form is at preventing the release of any part of the waste. So because the waste is radioactive, performance data is vital to determine whether or not the specific waste form is engineered so that it can contain the waste safely for over 1,000 years from now. The performance data can be used to model how much of the waste is able to escape within a certain amount of time and under what conditions it will escape. So there are 2 entries for the notes section. There's first a general notes section, which essentially lets whoever the data entry user is report any discrepancies between the document and the database, because there undoubtedly will be some. And to most importantly, report anything that a researcher may find useful under the assumption that they have not read the paper. The other section is an other analyses section. And this section asks that the users who are reporting the data include any testing or experiments done that don't have a place in the database, but are listed in the paper. This serves multiple purposes, but the 2 most important ones are that it allows researchers to pinpoint papers with the data they are looking for, even if that data isn't explicitly included in the database. And the second one is that in the future, if certain tests are added to the database during expansions, then whoever's in charge of updating the database would be able to look at that column and easily tell which documents have the data rather than having to read through all of the documents all over again. So the other data entry option is a bulk data importing, and I recorded a small video example that I'll play as I'm going. So bulk data importing is intended for researchers who are actually doing the experiments. Essentially, after you submit a report or publication, the researcher would copy and paste all of their data over from their Excel files into the bulk data tool. Anything that they don't have in their experiments, they would keep blank, because the bulk data tool autofills everything that is blank. Then they would enter the report information and submit however many samples they have through the bulk data import tool. If the data passes the validation measures that are in the tool, then it's submitted to the database. And the bulk data importing tool is really important because it helps keep the database up to date once all of the pre-published literature is entered in the future. So it wouldn't be this continual process of having to wait for the papers to be published. So while there's not enough time to show all of the features of the database, there were 2 final categories that I wanted to talk about. The first being some of the data quality considerations I've included. As you go through the sample by sample data entry, based on what you enter, you get warnings that pop-ups and hints on how you should continue to enter the information. The most important one being circled above. It's a cell that checks if the listed DOI number for the paper you're entering already has samples associated with it in the database, and it tells you how many. If you're entering information from that paper, it gives you a chance to double check and make sure the samples you're about to enter aren't already entered into the database. And it's really important to help prevent duplicates of the data. Additionally, there are a variety of macros which submit the data for both the data entry types. And these macros run through certain checks with warnings for the users entering the data to give them a final chance to correct anything that may be wrong. And lastly, as already mentioned, one of the future intended uses for this database is to be the basis for a machine learning model in which you'd be able to enter in what waste you have and get resulting grout formulations that would potentially work for that waste. While a machine learning algorithm actually is a much larger project because it requires so much data, this database provides a place for that data to go and is really important in starting that collection process. Additionally, for the now, I've included a introductory visualization tool, which would allow researchers to start analyzing those potential trends as the data is being entered. In the example graph I have above, I've plotted the compressive strength, or how strong the waste form is, against different formulation options. And there's no obvious trend from the data right now, but it's a really good representation of just how much data will need to be inputted before you start seeing these trends. It's worth noting, however, that the red line represents the legal minimum for how strong the waste forms have to be legally allowed. And so that's 500. So all of the formulations right now are above that strength. At the time of the report, there were almost 90 sample entries, but because each report looks at different characteristics of the waste forms, of those 90 entries, only 11 have measurements. So that kind of tells you that there's a 1 out of 10 dilution factor that kind of happens with the data whenever you're looking at these trends. And then lastly, as the database becomes more useful, the hope is that it can go outside of PNNL and WRPS and serve as a central location, more platform-based for other labs across the nation, and expanding to get literature from other national laboratories. Lastly, I'd like to acknowledge and thank my two mentors, Dr. Sarah Saslow and Dr. Matt Asmussen, for their guidance in this project in the summer. I'd also like to thank David Swanberg and WRPS, who sponsored my project, gave feedback on the database, and are actually really excited to start using it. And lastly, thanks to the GEM program and STEM education here at PNNL, who worked together to host PhD interns like myself. I literally would not be here without them. And thank you to all my friends, coworkers, and family who came out to support me on today. Thank you. Any questions?
Wow, thank you, Ariana. That was that's very interesting. Great work there. Open it up for some questions. I think I have one here in the chat from Gary. Why was Excel used, and are other databases able to be used?
So Excel was used as part of that user-focused element. Essentially, whenever I came in, I had a variety of ideas for the types of relational databases we could use. And they kind of off the bat were like, we would like to use Excel. It's the most user-friendly option we have. And it's easy to share and to look at simultaneously across the PNNL network. And so that was really important to them. And it was basically with the idea that if they expand outside of the size that Excel can hold without being, cumbered down or slowed, they would then move on to another tool like Tableau or BigQuery, or even potentially Access in Microsoft, just something that gives them more room.
All right, Gary, hopefully that answered your question. And then also a question, could this database be extended to make web mobile applications in the future?
Theoretically, yes, but that's not really the intended purpose of it. It's more of so that the long-term goal anyway is more of that whoever's running the waste immobilization plant for the cement waste forms would be able to continually have it running for their chemistry. It would be less for individual users, or it would be less for like international users. It would be more on a set up computer that's connected to the overall monitoring system.
Perfect. And then one other question, did you use macros to pull the data automation?
Yeah, so essentially every function that I've added in that a normal database would be able to do, including like the search and retrieve tools, I base all of that in various macros throughout the database.
All righty, thank you. Daniel also has a question in there. Oh, well, no, he was just a comment. So he says, great presentation. So Ariana, you might want to check the chat box there. Great work. All righty, so moving on, we're a few minutes behind, but that's okay. We like hearing about what students are doing. Next up, Austin Wang. If you go ahead and unmute yourself and share your presentation, you can go ahead and start. again, while he's getting his presentation ready, if you could please take the post survey of the event. I believe it's in the chat box. If not, Becky will go ahead and submit, put it in the chat box. If you cannot stay for the rest of the event, please, we ask that you complete that for us. Austin, the floor is yours.
Thank you. Okay, hi everyone. My name is Austin Wong. I'm a rising senior in high school. I'm working under Dr. Siren Chen on the ExoSheds project, which is a project designed to advance watershed system science using machine learning. And so I'll be presenting on building machine learning models to predict river water quality. So it is essential to maintain the quality of our waterways. I have a picture here of some dead fish in a river, and that usually results from hypoxia or anoxia, and which is just low dissolved oxygen concentrations, and the fish can't breathe, so they die. And access to continuous long-term data is crucial to measuring dissolved oxygen and other important water quality variables. However, such data is not always available in every single waterway in the United States. Due to costs, chemical sensors are expensive and available issues. Sometimes they also have missing data, sometimes they don't have it at all. And so it would be beneficial to be able to construct a model to predict water quality measures from the ones we do have. We do have a lot of temperature data, that's water temperature, and we do have a lot of discharge data, which is how much water is going by per second. Here are the sites of interest in the Mid-Atlantic hydrological region. The Passaic River is a major river in New Jersey. It flows from Newark towards the Pennsylvania-New Jersey border. And the Chester River is a river that flows from the Chesapeake towards Delaware. It's slightly east of Baltimore. There are more rivers that I conducted analyses on, but we'll focus on these two in this presentation. So this is a time series of the nitrate levels in Passaic, which is the left, and Crumpton, which is the name of the Chester River site, in the right. And as you notice, the nitrate levels are fairly high, 6 milligrams per liter and 10 milligrams per liter. They're approaching the safety guideline limits for human consumption. And it's also very cyclical, so it goes up and down very rapidly. And we would like to know when it is high, and also when the nitrate levels are high, does it correlate with anything else? And so monitoring nitrate is very important. This is a CQ plot between discharge and nitrate. This is to examine if there is any correlation, any relationship between these two variables. And as you can see here, there is a relationship. It's a power law, but that's not particularly relevant to the point. The point is that there is a correlation. The same with Crumpton. It's a very similar correlation. And contrasting that with discharge with dissolved oxygen, there is little to no correlation here. As you can see, the scatter plot is messy, it's everywhere. Something interesting to note, though, is that the colder seasons, so winter, tend to have higher dissolved oxygen levels than the warmer seasons, such as summer. And this is due to the fact that dissolved oxygen saturation is highly dependent on temperature. So there isn't a very strong relationship between discharge and dissolved oxygen, but there is a stronger relationship between temperature and dissolved oxygen. Here's another graph of Crumpton. They might illustrate the temperature splits. So we're going to apply a random forest model to try and relate these dissolved oxygen and nitrate with discharge and temperature. A random forest works very well with continuous values, which is what we have. We have data taken at 15-minute intervals or 1-hour intervals. And it's also fairly straightforward to implement, so there's no training you have to do to understand how the model works. How the model works is it's consisted of decision trees. It's consisted of several hundred decision trees, depending on how many estimators you want. So the model creates a forest of decision trees. All these decision trees individually take in some data and they input a prediction, and the forest accumulates all that predictions into one singular prediction. In a regression analysis, it would take the mean of all the trees. In a classifier analysis, it would take the load. And in this case, we're doing a regression. So it would take the mean of all of these decision trees and output that as its prediction. For this task, we'll use Jupyter Lab. The Jupyter Lab environment is very interactive. It's very friendly towards these types of tasks. It has pandas, which is a package used to create and process data into a viewable data frame. It also has scikit-learn, which we will import the random forest regression from. It also works well with NWIS and the data retrieval package, which is to get data from the US Geological Survey. And everything is programmed in Python, so it's easy to read, and it's very easy to someone who's not familiar with the workings of the project as to what the code is actually doing. So here's what it's look like. Here is what it looks like. So as for the results, we see strong results coming from strong CQ relationships. So from the CQ plots, discharge had a very strong relationship with nitrate. And so if we're predicting nitrate solely from discharge, we get a very strong results. R squared is 0.78, which is a pretty good fit. We also noticed that temperature and nitrate did not have that as powerful of a relationship. And so their model performance is severely worse than if we were to predict nitrate from discharge alone. However, if we combine the two, the model performs better. The same pattern is here in Crumpton. And when analyzing temperature and dissolved oxygen now, we see the exact opposite is happening. Temperature is more correlated with dissolved oxygen. And so if we're predicting dissolved oxygen from temperature alone, the model performance is much better than discharge alone. But once again, using both discharge and temperature results in a better model performance. And here are the results for Crumpton. And so the key takeaway from all these results is that stronger CQ relationships will give stronger model performances in random forest regression, but having an additional variable, that additional parameter, will yield a better model performance in this case than if we were to use either one individually. And so here's a time series of the predicted versus actual. As we can see here, it captures seasonal trends pretty well. So it captures the overall pattern. It looks like it fits reasonably well. It does do a little bit worse on the outliers. So these high frequency noises here and here. But overall, it does predict the nitrate pretty well. So the value that comes from this is that now we can use this model to predict DO or nitrate levels in sites that don't have these measurements, but they have discharge and temperature measurements. It could be a gap year, or the site could have none of discharge or oxygen, but we can use this model to help predict dissolved oxygen or nitrate. Besides these results, I learned that Jupyter Notebook is very interactive and easy to use. This interactivity has helped me throughout this project easily diagnose any codes or any errors in my codes. And there were more errors and codes that didn't work than codes that did. So that was very nice. And also generates many visualizations to help see what the data looks like, what we're actually analyzing, what the model is actually doing, and how it performs. I would like to thank the members of the ExaSheds project for their guidance, especially my mentor, Dr. Simran Chen, Pin Shuai, and Pei Shijiang. And also thank you to the people at the internship program office for organizing everything. Thank you. And now I'll open the floor to questions.
Thank you, Austin. Great work. Very impressive for a rising, you said you were a high school student. Are you graduating this year?
I'm a rising senior, so yeah, I'll be graduating.
Okay. Wonderful work. We'll open it up for questions. We are running a little behind, so if you could unmute yourself and ask a question, that'd be great. If not, you can always put it in the chat box.
Yeah, I got a quick question, Austin. So can you extend this model to other measures, such as maybe looking at phosphate levels in water? Like, are you guys thinking of extending this model to that?
So you could, but phosphate measurements are very rare. Not a lot of these sites carry them. And phosphate levels are also highly correlated with nitrate. So you could theoretically use this model to predict phosphate levels as well. But we haven't tried that yet.
Cool. All right, thanks. Great presentation.